Hey guys, so last week we had a class on mammalian cleavage. So these are how mammalian cells undergo cleavage right after fertilization. And I wanted to give a brief summary of that class by talking you through the images that we produced on the board. So remember, cleavage refers to the early cell divisions that occur right after fertilization. And in mammals, it begins on about day 1. Now, just to kind of fully summarize where all this occurs, mammalian cleavage begins right after fertilization. And what we have here is we have a brief or, a pretty simple sketch of the female womb. This brown oval, that's the ovary that released the egg into the ampulla of the oviduct. That happened around day 0. By day 1, cleavage is beginning, and cleavage is going to continue until that embryo emerges into the uterus around day 5. So day 5 is pointing to that little blue dot, which represents the embryo emerging into the uterus, where ultimately it will then undergo implantation, where that embryo will grow for the next 8.5 months or so. Okay, so let's go over here. Day 2, this is what the embryo looks like. You're still going to have that zona pellucida, which protects and covers the embryo. Remember that zona pellucida is a matrix of glycoproteins. It's like this thick membrane that protects the embryo from the outside. Its main job right after fertilization was to prevent other sperm from fertilizing the oocyte after the first sperm was able to do that. So what you'll see, this is after the first cell division or after the first cleavage. You have 2 cells and that small little blue dot. That's just the last polar body that's left over from meiosis of the oocyte. All right, now during this time at day 2, that embryo is in the oviduct, and cilia that line the inner wall of the oviduct are going to propel this embryo towards the uterus. That's how it kind of moves in the right direction. By the end of day 2, we've had our second cleavage, and now this is going to produce 4 cells. Now remember, mammalian embryos undergo rotational cleavage, which means that one cell is going to divide in one direction and the other cell divides in a completely different plane or direction. Also, these cell divisions don't occur at the same time. They can occur at completely different times, which can produce odd numbers of cells at any time during the first couple of days of cell divisions. By day 3, we're looking at around 8 cells, and this is when the proteins of the zygote are activated. And what that means is that before day 8, the cell divisions were really controlled by the messenger RNA and the proteins that were produced by the original oocyte or the egg. And now there's this transition where the proteins produced by the cells of the actual embryo are controlling cleavage. So it's kind of like the first time in which this embryo is controlling its own cell division, its own destiny in a way. Now, after this 8-cell stage, the embryo is going to undergo a process called compaction. This is when the cells on the outside are going to seal together with tight junctions. Remember, tight junctions are just these really strong cellular unions that prevent anything from moving in between the cells. And inside those 8 cells that are sealed together, you're going to have the cells on the inside. These are going to be protected by those outer cells, but they're joined together with gap junctions, which allow them to kind of trade ions and nutrients with each other. Gap junctions are like these little tunnels that exist between cells. Now, after this compaction process, this embryo of 8 cells is then going to be more tightly packed together. It kind of like shrinks in size within the zona pellucida. And these little black hash marks indicate that they're joined together with these really strong, tight junctions, right? By day 4, another cell division has occurred to produce a 16-cell embryo called a morula, all right? These 16 cells consist of 8 cells on the outside. These 8 cells on the outside will ultimately become the trophoblast cells, while the 8 cells on the inside, they're going to become the cells of the inner cell mass. From now on, everything in purple is going to represent cells that have derived from the trophoblast, while lime green represents the cells of the inner cell mass. All right. By day 5, we have a blastocyst that has finally formed. This is what the blastocyst looks like. We still have the zona pellucida as this blue outer covering. But now you have these trophoblast cells on the outside. That's what's in purple. And on one side of the trophoblast, you have a collection of green cells. These are the cells of the inner cell mass. This white area that's also within the trophoblast cells is the blastocele. This is a pocket of fluid that was formed when the trophoblast cells pumped sodium into this space. And we know that water follows salt, so water is going to follow the sodium to produce this blastocele, which is a fluid-filled cavity. All right. And so that really kind of describes the first 5 days of cell divisions. The next step for this blastocyst is for it to implant into the wall of the uterus, because remember, by day 5, it has emerged into the uterus. Just to kind of summarize that one more time, by day 5, here it is, into the uterus where it's going to undergo implantation. So next, we'll talk about how implantation occurs.